otherwise I can do the entire research on this. So I'll just tell you what is needed by you. Okay, so आपने याद रखना है जो C4 pathway है, this is also called as, और क्या कहते हैं इसको, Hatch and Slack pathway. This is also called as Hatch and Slack pathway. So who discovered C4 pathway? Hatch and Slack, यही बोलो, तभी तो पूछ रहा हूं मैं आपसे ज्यादा चालाक हूं. आप कौन स्मार्ट है, आपकी मैं?
आप सर.
चलो, आपने मुझे अभी देखा है. वैसे तो स्मार्टनेस बाहर से नहीं होती, अंदर से होती है. ठीक है, आप मुझे सुनते हो, आप मुझे जानते हो, दैट्स व्हाई आई एम स्मार्ट. सो लेट अस हैव दिस C4 pathway, and let me tell you, my friends, C4 pathway was first discovered by a scientist whose name is H.P. Kotsik. H.P. Kotsik. ये आप कभी पढ़ोगे नहीं ये सब, लेकिन I'm telling you, this was first discovered by a scientist called as H.P. Kotsik, where in sugarcane. In sugarcane. ठीक है. ये जो set of reactions है, the set of reactions were given by Hatch and Slack. तो ये जो set of reactions हैं, these reactions were, ये जो pathway है, the pathway was given by Hatch and Slack, but it was actually discovered by H.P. Kotsik in sugarcane. तो पहली चीज तो ये क्लियर हो गई हमारी. ठीक है. दूसरी चीज आपने याद रखनी है कि जो C4 pathway है, C4 pathway, this is found in 19 different angiospermic families. कितनी families में? This is found in 19 different angiospermic families. Is this very clear? ठीक है. तो 19 different angiospermic families have the C4 pathway. And out of these 19 families, let me tell you, out of these 19 families, ठीक है, we know कि most of these families, you know, they are basically, और ऐसे मत लिखो आप, ऐसे लिखो कि 19 families में से, चलो ये तो आप याद रखो कि 19 families, लेकिन आपने ऐसे सोचना है कि ऐसे मैं एक स्टेटमेंट दे रहा हूँ 75%, मैं स्टेटमेंट दे रहा हूँ कि 75% of all the C4 plants, ऐसे लिखो 75% of all the C4 plants are monocots. ठीक है. तो 75% of all the C4 plants are monocots. That means only 25% are dicots. Only 25% are dicots. 75% of the C4 plants are represented by monocots. तो मैं फिर से बना देता हूं. C4 plants, 75% are monocots, only 25% are dicots. इतनी बात समझ में आ गई? अब देखो, ये जो 75% monocots हैं, these are only represented by two families. मतलब 75% of all these C4 plants are represented by two families. याद रखो कि 75% of all these C4 plants which are monocots are only represented by two families. अब आप मुझे बताओ कौन सी हैं दो फैमिली? एक तो आप बता सकते, दूसरी नहीं बता सकते. Tell me which are these two families?
Poaceae.
ठीक है. So one is Gramineae, मतलब grass family. This is grass family. पहले हम कहते थे Gramineae, अब हम इसको क्या कहते हैं? Now we call this family as Poaceae. Poaceae. ठीक है. जैसे हम जो भी खाते हैं, हम गेहूं खाते हैं, हम भुट्टा खाते हैं, ठीक है, हम पल्सेस खाते हैं, दे ऑल, सॉरी नॉट पल्सेस, जितना ये गेहूं हो गया, आपका व्हीट हो गया, आपका ये राइस हो गया, ठीक है, ऑल दीज यू नो दे बिलोंग टू ग्रामिनी, दे ऑल ग्रासेस. जैसे आपका मेज हो गया, आपकी शुगर केन हो गई, दे ऑल, आपके जितने मिलेट्स हो गए, सो मिलेट्स, शुगर केन, ठीक है, आपका मेज, दे आर ऑल फ्रॉम ग्रामनी फैमिली, दे ऑल बिलोंग टू ग्रामनी फैमिली, दे शुड बी वेरी वेरी क्लियर। ठीक है, सो दो फैमिली है, वन तो ग्रामनी होगी, दूसरी कौन सी होगी, विच इज द सेकंड फैमिली, विच इज द सेकंड फैमिली, द सेकंड फैमिली इज कॉल्ड एज Cyperaceae. The second family is called as Cyperaceae. Keep this in mind. And the members of Cyperaceae are called as sedges. Members of Cyperaceae are called as sedges. So 75% of all these C4 plants which are monocots are only represented by two families. One is Gramineae, one is Cyperaceae. Okay? Now let us see the dicots. Let us look at the dicots. आपने याद रखना है, if I take this 25% as 100%, अगर मैं कहूँ all dicots, if I say all dicots which are C4, that means it becomes 100%. So out of all the dicots, we know कि 50% of dicots which are C4, याद रखो, 50% of all dicots which are C4 are only represented by a single family. They are only represented by single family. They are only represented by a single family. Keep this in mind. And which is this family? Can you tell me? कितना बड़ा फिगर हो गया कि 50% of all the dicots are only represented by a single family. And what is that? That family is Amaranthaceae. That family is Amaranthaceae. याद रखो. ठीक है? So this analysis is important. This you should keep in mind. Is this clear? And some of the common C4 plants, they are maize, millets. We have sorghum. We have sugarcane. We have atriplex. So these are some of the common C4 plants you should keep in mind. Okay. Now, अब देखो, let us come back to this C4 pathway. The question is, let us start with C4 pathway. See, all those plants which are growing in arid and semi-arid zones, याद रखो, all those plants which are growing in arid and semi-arid zones, we know daytime temperatures are very high. पता है ना आपको? Daytime temperature is very high. और मैंने आपको क्या बताया था कि during daytime temperature is high, what will happen to solubility of gases?
Decreases. Carbon dioxide.
Solubility of gases will decrease, and comparatively, what will happen to solubility of CO2? CO2 will also decrease. Solubility of CO2 decreases in the aqueous medium. And we know already the concentration of CO2 in the atmosphere is 0.034%. We know the amount of CO2 in the atmosphere is 0.034%. So at higher temperatures, this will further decrease. And what will happen to Rubisco? Rubisco will always function as, Rubisco will start functioning as oxygenase during the daytime. This will start functioning as oxygenase during the daytime. So that means there's a major problem in all those plants which grow under arid and semi-arid zones. That means during daytime they cannot photosynthesize. Photosynthesis कैसे होगा? How they will fix carbon? क्योंकि during daytime we know Rubisco mostly functions as oxygenase and not be able to capture the CO2. So in these plants, in all these plants, they have evolved a strategy. इन्होंने एक strategy evolve करी. They have evolved a strategy. और क्या strategy है देखते जरा. To take care of this Rubisco, to take care of this oxygenase activity of Rubisco, they have evolved a strategy. जब बच्चा कोई बहुत बदमाश होता है तो क्या करते हैं हम? मम्मी ने क्या किया था? बहुत बदमाशी करते थे छोटे में. कमरे में बंद किया, नहीं किया? पिटाई लगी और बंद कर दिया कमरे में. आप बंद हुए नहीं हुए? बताओ मुझे.
यस सर.
मेरे को तो बहुत बंद किया है. सो जब आप बचपन में बहुत ज्यादा बदमाशी करते हो तो मम्मी क्या करती है? जाके कमरे में बंद कर देती है. तो what do we do here? What we have done in these plants, the strategy is in arid and semi-arid zones, these plants, what we have done is we have confined Rubisco to a compartment. We have confined Rubisco to a cell inside a compartment, and this compartment has thick walls. We have confined Rubisco to a compartment, and what we do now in this compartment, we artificially maintain high CO2 concentration. We maintain high CO2 concentration. So we confine this Rubisco to a compartment, and here we maintain high CO2 concentration. अब क्या होगा? Will this Rubisco function oxygenase or carboxylase?
Carboxylase.
In the compartment. मम्मी जब कमरे में बंद कर देगी अपने आप से तानियां दूर हो जाएंगी. You will start functioning as carboxylase. जब कान टेढ़ेगी मम्मी ने. Now the question is, I'm just telling you, we have confined Rubisco to a compartment and artificially maintain high CO2 concentration. How do we maintain this CO2 concentration? How this high CO2 concentration is maintained? आपने याद रखना है, the CO2 concentration is maintained in this compartment by, मैं क्या कह रहा हूँ? How do we maintain this high CO2 concentration? We maintain it through मैं क्या कह रहा हूँ? We maintain this high CO2 concentration through a set of carboxylation and decarboxylation reactions. ये देखो, मैंने कहा कंपाउंड A है, ठीक है? ये क्या किया? ये हमारा CO2 CO2 comes, this is converted to B, this is converted to B. What happens? This B comes inside the compartment. ये यहाँ आया, B comes here, B is again converted to A. So what comes out now? CO2 comes out or not?
CO2.
And this A will again move back, again moves back. So how do we maintain this high CO2 concentration? We maintain this high CO2 concentration in this compartment. This is maintained by a set of carboxylation and decarboxylation reactions. Is this clear or not?
Yes, sir.
Okay, so we maintain it through a set of carboxylation and decarboxylation reactions. And my friend, let me tell you now, for your surprise, ये जो set of carboxylation and decarboxylation reactions हैं, this is called as the C4 pathway. The set of carboxylation and decarboxylation reactions is called as C4 pathway. और ये जो compartment है, this compartment has Rubisco plus other enzymes of C3 pathway. So Rubisco plus other enzymes of C3 pathway are maintained in the compartment. They are confined to the compartment. And artificially, what we are doing? Artificially, we are concentrating the CO2 in this compartment through a set of what? Through a set of carboxylation and decarboxylation reactions. Okay? So what is C4 pathway? C4 pathway is nothing else. C4 pathway is just a set of carboxylation and decarboxylation reactions whose sole purpose is what? Whose sole purpose is to concentrate CO2 in this compartment where Rubisco is confined. फिर से बोल दूं?
Yes,
आ
sir.
गया? Did you understand or not?
Sir, एक बार repeat कर.
मैंने ये बोला, what is C4 pathway? C4 pathway is nothing else. It is just a set of carboxylation and decarboxylation reactions whose sole purpose is what? Whose sole purpose is to concentrate CO2 in this compartment where Rubisco and other enzymes of C3 pathway are confined. Did you get it now?
Yes, sir.
Okay? So now we know what is C4 pathway. C4 pathway is nothing else. It is just a set of reactions whose sole purpose is to concentrate CO2. Or I can say like this, what is C4 pathway? C4 pathway is nothing else. It is a CO2 concentrating mechanism. Right or wrong?
Yes, sir.
यही तो है. What is C4 pathway? It is nothing else. It is a CO2 concentrating mechanism. बस खत्म. अगर एग्जाम में कोई पूछ ले what is C4 pathway, तो इतना लंबा चौड़ा लिखने की जरूरत ही नहीं है आपको. C4 pathway is nothing else. It is a CO2 concentrating mechanism. Nothing else. Through a set of carboxylation and decarboxylation reactions. पहले तो बात ही समझ में आई कि नहीं आई? रट्टा नहीं लगाना है, नहीं तो आप लोग कॉलेजेस में रट्टा लगाना चालू कर देते हो बस. ठीक है, तो अब तो दैट मीन्स मैंने आपको अभी बताया था कि C4 प्लांट्स आल्सो हैव व्हिच पाथवे, दे आल्सो हैव C3 पाथवे, बताया था नहीं बताया था?
Yes, sir.
अब बात समझ में आई कि why these plants have C4 pathway? They already have C3 pathway also, but the function of C4 pathway is just to concentrate the CO2. So now what will happen? Now most of the times the rubisco will function as carboxylase. That means the sole purpose is to reduce photorespiration. This will reduce what? This will reduce, क्योंकि जब rubisco will function as carboxylase, so it will not function as oxygenase. So it will not function as oxygenase मतलब क्या है? This will reduce photorespiration. This will reduce photorespiration. So there's a reduction of photorespiration. मतलब ये जो C4 plants हैं, they're highly efficient. They are more efficient than C3 plants. याद रखो, C4 plants are more, मैं कह सकता हूँ ना कि C4 plants are more efficient than C3 plants in fixing CO2. They are more efficient than C3 plants in fixing CO2. But it is at a cost. It is at a certain cost. There's a certain cost involved. We see this later. We know there's a certain cost which is involved. We'll see this later. अभी हमें मोटा मोटा पता लग गया कि भैया ये क्या होता है C4 pathway. Nothing else, my friend and countryman, it is a CO2 concentrating mechanism. That is it. अब हमने क्या पढ़ना इस पाथ में? अब रह क्या गया? You understand the soul. अब आपने जब मेरा आत्मा को देख लिया तो बचे ही क्या? अब तो बस हमें रिएक्शंस देखने हैं बस कि which are these reactions, nothing else. ठीक है, so let us look at this now. अब एक चीज और याद रखो जरा, ये जो C4 प्लांट्स होते हैं, C4 प्लांट्स की जो लीव्स होती हैं, ये देखो, ये मैंने कहा कि एक C4 प्लांट की लीफ है, ठीक है, इनकी जो लीव्स हैं इसकी एक स्पेशल एनाटमी है. अगर आप इसका सेक्शन काटोगे, इसका आप टीएस काटोगे, तो यू विल सी दे हैव अ स्पेशल एनाटमी, ठीक है, एंड वी कॉल इट क्रैन्स एनाटमी. सो देयर लीव्स हैव अ स्पेशल एनाटमी, वी कॉल इट क्रैन्स इज अ जर्मन वर्ड, क्रैन्स एनाटमी. They have anatomy which is called as Kranz anatomy. अब देखो क्या होता है यहाँ पे क्या हो रहा है, ये देखो बीच में क्या वैस्कुलेचर है हमारा, ये वैस्कुलेचर है हमारा, यहाँ पे हमारा जाइलम है, यहाँ पे हमारा फ्लोएम है, वैस्कुलेचर होता है, ठीक है. C4 plants में we see that around the vasculature we have thick walled cells. Vasculature is surrounded by thick walled cells. This vasculature is basically surrounded by special thick walled cells, and we call these thick walled cells as bundle sheath cells. We call them as bundle sheath cells. So vasculature is surrounded by thick walled cells, we call them as bundle sheath cells, ठीक है. और ये जो bundle sheath cells हैं, these bundle sheath cells are in touch with another set of cells. देखो, ये देखो, they're in touch with another set of cells. देख रहे हो आप, bundle sheath cells are in touch with another type of cell, and what is this cell? We call it mesophyll cell. We call it mesophyll cell. So there are two types of cells, one is mesophyll cell, one is bundle sheath cell, ठीक है. और आपने देखा है कहीं मैंने दो mesophyll cell नहीं बनाए, हर जगह एक ही एक बनाया है. मतलब कहने का कि ये जो mesophyll cells हैं, none of these mesophyll cells are one cell away from the bundle sheath. हम ऐसे लिखते हैं इसको कि they're not, मतलब एक ही सेल है बस, ये सिंगल सेल. यहाँ पे हमने कभी दो नहीं बनाए, ऐसे करके दो नहीं बनाए मैंने, ठीक है. तो each bundle sheath cell is in direct touch with the mesophyll cell, ठीक है. एंड वी नो ये हमारा स्टोमेटा है, ये देखो ये स्टोमेटा है यहाँ पे, दिस इज अ स्टोमेटा हो गया, दिस इज द स्टोमेटा. दैट मींस व्हिच सेल्स आर इन टच विद द एटमॉस्फेयर? mesophyll cells. Mesophyll cells are close to the atmosphere and bundle sheath cells are away from the atmosphere. ये देखो, जो bundle sheath cells हैं, they are away from the atmosphere. Mesophyll cells are near the atmosphere, and these bundle sheath cells are thick walled. We know they are thick walled. और आपने याद रखना है कि जो bundle sheath cells हैं, they are also coated with a waxy layer. They are coated with a waxy layer which is called as suberin. They also have a layer of suberin. They have a layer of suberin also, which will not allow the gases to enter or leave. This is a waxy layer. So gases are not allowed to enter or leave. ठीक है, तो look at this kind of anatomy over here. ठीक है, और आपने याद रखना है कि both these cells, mesophyll as well as bundle sheath cells, both have chloroplast. Both these cells have chloroplast. Both these cells also have chloroplast. यहाँ पे भी chloroplast है और यहाँ पे भी chloroplast है. तो mesophyll cells also have chloroplast and bundle sheath cells also have chloroplast. Is this clear? अब आपने देखा है, now look at this. हमने क्या देखा यहाँ पे? हमने देखा कि the vasculature is surrounded by a ring of cells. Don't you see? This vasculature is surrounded by a ring of thick walled cells. मतलब ये जो cells हैं, ये जो bundle sheath cells हैं, they form a kind of a ring. They form a kind of a halo. Halo मतलब जो भगवान जी के सर के पीछे रोशनी होती है, नहीं होती है? जब मंदिर में जाते हो तो मंदिर में देखते हो ना कि भगवान जी के सर के चारों तरफ रोशनी सी आ रही है. There's a kind of a halo. This is called as a halo. Is this very clear? और आपने याद रखना है, मतलब there's a halo around the god's head, just like bundle sheath cells. And this halo in German is called as Kranz. This halo, this ring, is called as Kranz in German. And that is why we call this anatomy as Kranz anatomy. That is why we call it Kranz anatomy. Is this clear or not?
Yes, sir.
Yes, sir.
हाँ जी, boys are near to me क्योंकि कम है. They are more closer.
Yes,
ठीक है? So is this very clear? Why do we call it Kranz anatomy? And what is the special anatomy over here? So we have bundle sheath cells, we have mesophyll cells. ठीक है? अब ये देखो एक चीज मैंने अभी क्या बताया था. What is C4 cycle? A set of carboxylation decarboxylation reactions. देखो जरा. तो मैंने कहा ये देखो, ये हमारा mesophyll cell है, ये हमारा bundle sheath cell है, और रुबिस्को कहाँ confined है? रुबिस्को कहाँ पे है? रुबिस्को is found in the bundle sheath. जो C3 cycle के enzymes हैं, they are found in the bundle sheath cells. So all these C3 cycle enzymes are confined to the bundle sheath. बात समझ में आ गई? यही तो हमने देखा था पहले कि रुबिस्को is confined to a compartment. Which is that compartment? That compartment is nothing else, that is bundle sheath. अब क्या होगा देखो, CO2 will enter. ये A आया, A is converted to B, CO2 enters, B comes over here, converted to A goes back, CO2 comes out. तो क्या हो रहा है? This is C4 pathway. That means ये जो C4 pathway वो कहाँ start हो रहा है? C4 pathway basically starts in the mesophyll. Are you getting my point or not? The C4 pathway basically starts in the mesophyll cells. The first reaction of
Yes,
C4
sir.
pathway will take place in bundle sheath and mesophyll. बोलो.
Mesophyll
Mesophyll.
cell.
And which cells are closer to atmosphere?
Mesophyll cell.
Mesophyll. Is this very clear? So C4 cycle is nothing else. It is a set of carboxylation and decarboxylation reactions. Sole purpose is to concentrate CO2 in the bundle sheath cells, where all the enzymes of C3 pathway are concentrated. ठीक है, बाकी सब तो कहानी है. अब देखो, मैंने कहा Kranz anatomy. Look at this. भाई ये हमारा एक mesophyll cell है.
जो
हमारा
क्लोरोप्लास्ट आपने
क्या करें?
सर जो क्लोरोप्लास्ट आपने मेसोफिल सेल पे बनाया है वो C3 ही करेगा.
नहीं, mesophyll cells में C3 नहीं होगी यहाँ पे. ठीक है, याद रखो, mesophyll cells only
नहीं
have
सर,
C4.
मेसोफिल सेल्स पे जो आपने क्लोरोप्लास्ट अटैच कराया था, आपने कहा था ना कि बंडल शीट और मेसोफिल
क्लोरोप्लास्ट है
सेल
वहां पे,
दोनों
लेकिन
में.
उसमें C3 के एंजाइम्स नहीं है.
ओके सर.
So chloroplast here will lack the enzymes of C3 pathway. Is this very clear? In mesophyll
Yes.
cells. तो देखो यहाँ पे एक चीज देखो, यहाँ पे मैंने कहा ये हमारा वैस्कुलेचर है, ये हमारा वैस्कुलेचर है यहाँ पे, जाइलम फ्लोएम, ये हमारा बंडल शीथ सेल है, ये हमारा मेसोफिल सेल है. ठीक है, और ये जो और ये हमारा देखो, ये हमारा एपिडर्मिस है, लीफ का एपिडर्मिस, यही तो है एपिडर्मिस. लीफ का। यहाँ पे स्टोमेटा है, देखो स्टोमेटा, ये अपर एपिडर्मिस हो गया हमारा, यहाँ से CO2 एंटर्स, CO2 अंदर आ गई यहाँ पे मेसोफिल सेल में। अब देखो हाउ बंडल शीथ सेल इज कनेक्टेड टू मेसोफिल, दे आर कनेक्टेड बाय मींस ऑफ, ये देखो देयर इज अ कनेक्शन हियर and they are connected by means of plasmodesmata. This is what? This is plasmodesmata. So mesophyll cell is connected to bundle sheath by means of plasmodesmata. Is this very clear? Now we know we have a Cran's anatomy. We have a Cran's anatomy. Do you think there's some importance of this anatomy? There's a significance of this anatomy? Let us see some importance of Cran's anatomy. Nothing happens without a reason. There must be some importance of Cran's anatomy. So what is the importance of Cran's anatomy? If I ask you a question, what is the importance of Cran's anatomy? आपने याद रखना है, the first importance is that there can be a division of labor. There can be a division of labor between the two types of cells. There's a division of labor between the two types of cells. ठीक है? Who will, यहाँ पे क्या हो रहा है? Who captured CO2? Tell me, which cell captured CO2?
Mesophyll.
You know, mesophyll cell will capture the CO2, and which cell will fix the CO2?
Bundle.
Now you understand what is the difference between capture and fixation. Fixation. So capture is taking place in mesophyll, but the fixation of CO2 will take place in bundle sheath. Is this very clear or not? So that means there's a division of labor between the two types of cells, and it is possible because of Cran's anatomy. दूसरी चीज देखो, दूसरा इंपॉर्टेंस ये है, ये जो बंडल शीथ सेल है, we know bundle sheath cells have thick wall. You know these cells are basically thick walled or not? We know bundle sheath cells are basically thick walled cells, and they're also away from the atmosphere. The thick walled cells, they're away from the atmosphere, and thus they can maintain high CO2 concentration. High CO2 concentration can be maintained in these cells क्योंकि इनके walls are thick, they have suberin, so CO2 will not move out. So that means high CO2 concentration can be maintained. That means reduction in photorespiration. There's a reduction in photorespiration, and that is also possible because of Cran's anatomy. This is possible because of Cran's anatomy, that we can eliminate photorespiration. We can nearly eliminate photorespiration. Is this clear? अब तीसरी इंपॉर्टेंस देखो भाई, ये देखो C3 साइकिल कहां हो रही है? हमें पता है जो C3 साइकिल है दैट इज टेकिंग प्लेस इन बंडल शीथ. तो C3 साइकिल से कुछ प्रोडक्ट तो निकलेगा. C3 साइकिल से क्या हो जाएगा? यहां से हमारा सुक्रोज बनेगा, यहां पे स्टार्च बनेगा. अब रूट्स कह रही है भाई मुझे भी खाना चाहिए, ये तो लीव्स में हो रहा है ना, दिस इज ऑल टेकिंग प्लेस इन लीव्स. ना रूट्स सेइंग कि भाई मुझे भी भूख लगी है, सो क्या करोगे? ट्रांसपोर्ट करोगे नहीं करोगे? और किससे के थ्रू ट्रांसपोर्ट होता है? ट्रांसपोर्ट होगा वैस्कुलेचर के थ्रू. तो वैस्कुलेचर इज फार ऑफ कि नियर बाय? पापा आपकी फैक्ट्री कहाँ खोलेंगे? जहाँ रेलवे स्टेशन 50 किलोमीटर दूर है या 100 किलोमीटर दूर है? या पापा फैक्ट्री वहां खोलेंगे जहाँ 2 किलोमीटर दूर है रेलवे स्टेशन?
2 किलोमीटर.
तो फिर दिस इज ऑल्सो वन ऑफ द बेनिफिट्स. दैट मीन्स ये जो बंडल शीथ सेल्स हैं, बंडल शीथ सेल्स आर इन क्लोज कॉन्टैक्ट विद द वैस्कुलेचर. We know bundle sheath cells, they are in close contact with the vasculature, so it becomes easier to transport photoassimilates. Or simply food बच्चों के लिए. So it becomes easier to transport the photoassimilates. So this is also a significance of the Cran's anatomy. Is this clear or not? And we know, according to an analysis, we know कि the concentration of CO2, याद रखो, concentration of CO2 in the bundle seed cell, the concentration of CO2, the concentration of the CO2 in the bundle seed cells is 10 times higher. This is 10 times higher than that in the atmosphere. याद रखो, मतलब bundle seed cells can maintain 10 times the concentration of CO2 in comparison to the atmosphere. Is this clear or not? इतनी बात समझ में आई कि नहीं आई?
Yes.
ठीक है, अब देखते हैं यहाँ पे, देखो. Now let us look at the C4 cycle. Let us look at the pathway. अब reactions देखते हैं, और तो कुछ बचा नहीं इसमें. Let us look at these reactions over here. ठीक है, अब देखो ये reaction. पहले हम बनाते हैं यहाँ पे mesophyll cell. You take a new page. Take a new page over here and look at this now. This is a mesophyll cell, and we know this is a bundle sheath cell. This is a bundle sheath cell. This is a mesophyll cell. This is the epidermis. This is the stomata. ठीक है. So look at this over here. So look at this over here. Just hold on. Let me shift this. Okay. So we know now. This is the. So now what happens over here? This is the mesophyll. And this is the bundle sheath. ठीक है. अब देखो, इनके क्लोरोप्लास्ट भी हैं दोनों में. ये देखो, ये आपने बहुत, this should be very very clear. This is the chloroplast of mesophyll. And this is the chloroplast of bundle sheath. अब कौन सी रिएक्शन कहाँ हो रही है? You should keep this in mind. This is the chloroplast. This is also the chloroplast. ठीक है. Now let us see this. Let us see this now. CO2 enters. This is the cytoplasm of mesophyll cell. This is the cytoplasm. So CO2 enters, reaches the cytoplasm of mesophyll cell. In the cytoplasm of this mesophyll cell, this CO2 is combined with water. CO2 is combined with water through an enzyme which is called as carbonic anhydrase. What is the enzyme? The name of the enzyme is carbonic anhydrase. And what we get?
Carbonic
carbonic anhydrase. We get H2CO3, and this H2CO3 is converted to SCO3 negative. That means finally what we get is bicarbonate ions. So through this capture, what we finally get is bicarbonate ions. Is this very clear? तो मैं मिटा देता हूँ ये, क्योंकि I have less space, but आपको याद रखना है ये. ठीक है? So they will combine here to give us HCO3 negative through an enzyme called as carbonic anhydrase. ठीक है? So this is, so we get SCO3 negative. Now look at this. Just hold on. Yeah. So let us see the first reaction now. The first reaction here is PP. So who's the acceptor of this carbon? The acceptor of this carbon is PP, phosphoenol pyruvate. So everything starts with PP, phosphoenol pyruvate. Okay? This phosphoenol pyruvate will capture this SCO3 negative. This will capture this SCO3 negative. PP is 3 carbon compound. SCO3 negative has 1 carbon. And this reaction is catalyzed by, this reaction here, this is catalyzed by an enzyme which is called as PEP carboxylase. What is the name of the enzyme? This is PEP carboxylase. This is the enzyme, PEP carboxylase. And finally, what do we get? Finally, we get a 4 carbon compound, which is oxaloacetic acid. So this is the first reaction. And the first compound which is formed is oxaloacetic acid, which is 4 carbon compound. And that is why we call it C4 cycle. Because the first compound which is formed is a 4 carbon compound. So we call it what? We call it C4 cycle. So this should be very clear that the substrate for this enzyme is not CO2. The substrate for this enzyme is bicarbonate ions. Lekin substrate for Rubisco is CO2. Is this very clear? ठीक है, अब देखो क्या होता है. ये जो oxaloacetic acid है, this oxaloacetic acid now in the next step it enters the chloroplast. ये chloroplast के अंदर आ गया. And in the chloroplast this oxaloacetic acid is converted to malate. This is converted to malate. ठीक है, आप हमेशा देखते हो जहां oxaloacetic acid है वहां malate बन जाता है. ऐसा क्यों होता है? Because oxaloacetic acid is unstable compound. याद रखो, oxaloacetic acid is comparatively unstable. So something which has to be transferred to bundle sheath has to be transferred in a stable form. Something which has to be transferred to bundle sheath should be more stable, and that is why oxaloacetic acid is converted to malate. This is very clear. And in the next step, चलो enzymes भी देख लेते हैं यहीं पे साथ साथ. This reaction here is catalyzed by an enzyme which is called as NADPH dependent malate dehydrogenase. This is NADPH dependent malate dehydrogenase. That means in this reaction, आपने याद रखना, in this reaction, one molecule of NADPH is converted to NADP positive. One molecule of NADPH is converted to NADP positive. And finally we get malate. We get malate. भाई जब malate से oxaloacetic acid बनता है, respiration याद नहीं है?
TCA साइकिल में.
TCA साइकिल में, हाँ, respiration जब TCA साइकिल में तो वहां यूज होता है. तो जब उल्टा आएगा तो क्या, यही तो हो रहा है यहाँ पे. वहां पे प्रोडक्शन होता है, यहाँ पे यूज हो रहा है. याद है ना? So this is what, this is NADPH dependent malate dehydrogenase. So finally we get malate. Now this malate is transported to the bundle sheath cells, enters the chloroplast. It enters the chloroplast of bundle sheath cell. ये malate यहाँ आ गया. 4 carbon compound. Malate comes over here. This is 4 carbon compound. ये देखो malate यहाँ पे आ गया. ठीक है, अब क्या होता है देखो. तो बंडल शीथ में तो आ गए हम. अब बंडल शीथ में क्या हो जाएगा? This malate which is 4 carbon compound is converted to a 3 carbon compound pyruvate. This is converted to pyruvate. Malate is 4 carbon. Pyruvate we know is 3 carbon. So what will happen to one extra carbon? One extra carbon comes out as CO2. CO2 is released over here. This is very clear. And now in the next reaction, this pyruvate moves back to the mesophyll cell, enters the chloroplast. ये pyruvate यहाँ पे आ गया. Pyruvate comes back. CO2 is released. The concentration of CO2 starts increasing in the bundle sheath. Concentration of CO2 starts increasing in the bundle sheath. And I have already told you कि यहाँ पे ये chloroplast है. This chloroplast has all the enzymes of C3 pathway. So now the CO2 would be captured by the C3 pathway. This is fixed now. यहाँ पे capture हुआ था. Now the CO2 would be used by the C3 pathway, and now it is fixed. Now it is fixed. And we get what? We get triose phosphate. We get triose phosphate is released. Pyruvate moves back to the mesophyll cell. And in the next reaction, ये देखो, in the next reaction, this pyruvate is converted to PP. In the next reaction, we know the pyruvate is converted to PP inside the chloroplast itself. And who catalyzes this reaction? ये देखो, very important enzyme. Who catalyzes reaction? This is catalyzed by PPDK. Pyruvate phosphate dikinase. This is pyruvate phosphate dikinase. ठीक है? Conversion of this pyruvate to PP. याद रखो, again important. Conversion of this pyruvate to PP in this reaction, one ATP is converted to AMP. याद रखो, in this reaction, one ATP is converted to AMP. So in this reaction, one ATP is converted to AMP. And we know, ये जो conversion of ATP to AMP, conversion of ATP to AMP, this is said to be equivalent to 2 ATP molecules. This is energy released is said to be equivalent to 2 ATP molecules. This is regarded equivalent to 2 ATP molecules. So ATP to AMP, this is equivalent to 2 ATP molecules. And finally, what is happening? Finally, we get PP. PP moves out into cytoplasm. ये देखो, PP moves out into cytoplasm. And again, ready to capture another carbon. Again, ready to capture another carbon. Did you get this reaction or not? So what is the C4 pathway? क्या है ये? नथिंग एल्स, इट इज अ सेट ऑफ, बताओ
कार्बोक्सिलेशन डीकार्बोक्सिलेशन
बस खत्म, यही तो है, और सब कहानी है, और तो सारी कहानी है. It is nothing else, it is a set of carboxylation and decarboxylation reaction whose sole purpose is what? The sole purpose is to concentrate CO2 in the bundle sheath cells, nothing else. And now the Rubiscol function is carboxylase. Photorespiration is eliminated.
सर यहाँ पे 2 ATP समझ में नहीं आया सर.
अभी आप इतना ही समझ लो कि कन्वर्जन ऑफ ATP, भाई ऐसा है, ये देखो ATP to ADP, ठीक है, और फिर ATP to ADP, चाहे आप ऐसे लिख लो, चाहे हम ऐसे लिख दें ATP to AMP. What we say, this is equivalent to this only. Conversion of ATP to AMP is regarded to be equivalent to this, and that is why we call it equivalent to 2 ATP molecules. Is this clear?
Yes.
इसको मैं आपको डिटेल में बता सकता हूँ. I can discuss this later with you, okay? कि why we call this. अच्छा, तो अभी हमने बात करी C4 pathway is nothing else, it is a set of carboxylation and decarboxylation reaction. अच्छा, मुझे एक चीज बताओ, यहाँ पे conversion of oxaloacetic acid to malate 1 NADH being consumed, और यहाँ पे malate to pyruvate CO2 is released, but this step not only releases CO2, but in this step we see कि in this step malate to pyruvate हमने क्या देखा है, यहाँ पे 1 space नहीं है यहाँ पे, I'll just write it over here. In this 1 NADP positive is converted to NADPH. तो मतलब ये जो रिएक्शन है malate to pyruvate, this not only releases CO2 but this also produces NADPH. This reaction also produces NADPH. ये भी याद रखो आप. And there is a significance of this. There's a significance of this. तो इतना आपको समझ में आ गया, नहीं आ गया?
Yes, sir.
बात समझ में आ गई? ठीक है. अब देखो, ये जो C3 साइकिल है, C3 साइकिल कहां पे हो रही है? बंडल शीथ सेल में हो रही है, क्लोरोप्लास्ट में हो रही है. ठीक है. अब देखो, आप एक चीज देखो, मैंने कहा क्लोरोप्लास्ट है बंडल शीथ सेल का, तो C3 साइकिल को क्या चाहिए? C3 साइकिल को क्या क्या चाहिए बताओ? C3 साइकिल को ATP चाहिए, और C3 साइकिल को
NADP प्लस चाहिए.
C3 साइकिल को ATP चाहिए, C3 साइकिल को NADPH चाहिए. ठीक है. तो मतलब C3 साइकिल को लाइट रिएक्शंस भी चाहिए, यहाँ पे लाइट रिएक्शंस भी होंगी, तभी तो NADPH बनेगा. और जब लाइट रिएक्शंस होंगी तो पानी टूटेगा कि नहीं टूटेगा?
यस सर.
ऑक्सीजन इवॉल्व होगी कि नहीं होगी? ऑक्सीजन इवॉल्व होगी कि नहीं होगी?
होगी सर.
तो बंडल शीथ सेल्स में तो मतलब अगर लाइट रिएक्शंस होंगे तो यहाँ पे ऑक्सीजन इवॉल्व होगी, फिर तो सारा पर्पस ही फेल हो गया हमारा. फिर द एंटायर पर्पस ऑफ कन्फाइनिंग दिस रुबिस्को टू कंपार्टमेंट फेल्स. क्योंकि अगर यहाँ पे फोटोसिंथेसिस होगा, if photosynthesis will take place over here, that means oxygen would be produced, and that is why आपने याद रखना है, ये जो C4 साइकिल है, C4 साइकिल not only transports CO2, it also transports reducing power in the form of NADPH. देख रहे हो, ये जो C4 साइकिल है not only transports CO2 to the bundle sheath, but C4 cycle also produces and it also transports reducing power in the form of, देख रहे हो, NADPH. That means now less NADPH would be produced by light reactions.
Yes sir.
Less NADPH has to be produced now. Less NADPH production मतलब क्या? Less breakdown of water.
Less right reactions. Yes sir.
Less breakdown of water, that मतलब क्या? Less production of oxygen. So this is very important कि ये जो C4 cycle it not only transports CO2 but it also transports reducing power. So we can reduce, so these bundle sheath cells can reduce the production of NADPH. The production of NADPH can be reduced. So less water has to be broken down. Less water has to be broken down. Is this very clear or not? Okay?
Yes, sir.
So we have talked about the C4 pathway, जो मोटा मोटा मैंने आपको बता दिया जो जरूरत है. अदरवाइज इसको डिस्कस करने के लिए आई नीड वन मोर लेक्चर. I require one more lecture to discuss C4 pathway, and that is beyond the scope. ठीक है, तो this you should remember. और मैं एक चीज आपको और बता देता हूँ कि we know there are certain plants, we know, मैंने आपसे क्या बताया था कि oxaloacetic acid which is produced here is unstable. This is unstable comparatively. That means it has to be transported in a stable form. मैंने ये बताया था आपको. Is it clear or not? So how they stabilized? Either we can reduce it to malate. ये क्या हो रहा है यहाँ पे? Oxaloacetate से malate reduction ही तो हो रहा है ये. भाई oxaloacetic acid से malic acid बन रहा है, क्या हो रहा है ये? यहाँ पे क्या हो रहा है? Reduction हो रहा है ना ये?
Yes, sir.
So how we can stabilize oxaloacetic acid? We can stabilize this either through reduction to malate. This is one way. What else? There's another method, you know, कि मैं नया पेज ले लेता हूँ. ये oxaloacetic acid है हमारा. Either this can be reduced to malate. यहाँ पे क्या हो रहा है? NADPH is converted to NADP positive. This is क्या? This is reduction. Or we see there's a second method here. What we also see, oxaloacetic acid can also be transaminated. This can be transaminated to form alanine. ये सब वहीं हो रहा है. सब कहाँ हो रहा है ये? Mesophil में. This all is in mesophil. So either certain plants, आपने याद रखना है कि certain C4 plants will produce malate and certain C4 plants produce alanine. That means we have variety of C4 plants also. We have 3 different subgroups of C4 plants, याद रखो. Classified on different bases. You don't need to know. But certain C4 plants are alanine formers, certain C4 plants are malate formers. मतलब कुछ malate transfer करेंगे bundle sheet में, कुछ alanine को transfer करेंगे bundle sheet में. Is this very clear? Sorry, don't write alanine. Aspartate, right? Aspartate, not alanine, aspartate. Some are malate formers, some are aspartate formers. Yeah, don't write alanine, aspartate. So some C4 plants will transport malate, some C4 plants will transport aspartate. So some C4 plants are aspartate formers, some C4 plants are malate formers. तो यहाँ पे देखो aspartate आ गया. चलो हम देखते हैं aspartate आ गया. ठीक है, और यहाँ पे क्या आया था? malate आया था. malate से क्या बना था हमारा? pyruvate. और यहाँ पे CO2 रिलीज हो गई थी. CO2 is released. जो aspartate formers हैं, aspartate formers on their decarboxylation will produce alanine. यहाँ पे alanine यहाँ बनेगा. We can get alanine, again CO2 is released over here. We get alanine. ठीक है, and this alanine can move back. यहाँ पे pyruvate वापस चला जाएगा, यहाँ पे alanine वापस चला जाएगा, and alanine, ये देखो alanine in mesophil can again be converted to pyruvate. तो end में हमें pyruvate ही मिलेगा. Finally we get pyruvate, whether it is pyruvate coming back or whether it is alanine coming back. Alanine would also be converted to pyruvate. So what I'm trying to tell you, we have different subgroups of C4 plants also. Some are aspartate formers and some are, कहाँ चला गया हमारा? Malate formers. ठीक है? So this concept should also be very, very clear. Now, let us see the energetics of C4 plants. How much energy do they need? भाई C4 plants में C3 pathway तो है ही. C4 plants also have C3 pathway. तो C3 pathway में to fix one CO2, how much is the cost?
So 3 ATP and 2 NADP.
CO2 and
3 ATP and 2 NADPH. This is the cost we know. Plus यहाँ पे C4 cycle भी हो रही है, C3 भी हो रही है, C4 भी हो रही है. अब C4 में क्या होता है? C4 में A to B, ये आ गए यहाँ पे B to A goes back. यहाँ पे क्या हो रहा था याद है? कौन सा एंजाइम था यहाँ पे? PPDK. ये एंजाइम था ना PPDK, पायरुवेट फॉस्फेट डाइकाइनेज. क्या कर रहा था? कन्वर्जन ऑफ ATP टू
AMP.
C4 cycle has a cost. C4 cycle also has a certain cost which is equivalent to 2 ATP. Which is equivalent to 2 ATP. That means to fix one carbon, to capture and fix one carbon, we need 2 more ATP. So C4 cycle की cost क्या हो जाएगी? C4 cycle के through if you have to fix one CO2, that would be equivalent to what? 3 ATP plus 2 ATP. This is what? 5 ATP plus 2 NADPH. So what is the cost of fixing one CO2 through C4 cycle? बताओ मुझे.
11.
11. 5 ATP हुआ ना. To fix one CO2, how much? 5 ATP.
5 ATP,
5
2
ATP
NADPH.
वही है C3 वाले, और ये 2
Yes,
ATP C4 वाले हैं.
हम्म.
तो टोटल कितने हो गए?
5 ATP and 2 NADPH.
So the cost of fixing one carbon is 5 ATP and 2 NADPH. Have you
तो
understood?
सर ये सारे C4 पाथवे में थोड़ी काउंट होंगे.
कौन से सारे नहीं काउंट होंगे?
मतलब जो 3 ATP है वो तो C3 पाथवे वाले हैं ना.
मैं तो
C4 में तो
C3
एडिशनल.
मैं तो C3 C4 की बात ही नहीं कर रहा हूँ. मैं तो एक कार्बन की कॉस्ट निकाल रहा हूँ बस.
ठीक है सर.
आप ही मुझे बता दो, एक कार्बन फिक्स करने की कॉस्ट क्या होगी? यही होगी कि नहीं होगी? मतलब द कॉस्ट ऑफ फिक्सिंग वन कार्बन इज व्हाट? 5 ATP and 2 NADPH. मतलब ये जो C4 पाथवे है, दिस इज मोर कॉस्टलियर.
यस सर.
That means
सर आपने बोला था कि C4 पाथवे में NADPH रेड्यूस करेगा, मतलब C3 पाथवे की.
हाँ, तो क्या प्रॉब्लम क्या होगी इसमें?
तो 2 NADPH ही होंगे या फिर कम होंगे?
इतने ही होंगे. C3 पाथवे को तो यही चाहिए ना, 2 NADPH. बस
Okay,
हमने
sir.
प्रोड्यूस ना करके हम पीछे से ले आए उसको.
Oh.
इज दिस वेरी क्लियर? कॉस्ट यही रहेगी इसकी. कॉस्ट यहाँ पे यही रहेगी, 5 ATP and 2 NADPH. तो कहने का मतलब ये हुआ कि इफ वी लुक एट दिस, टू प्रोड्यूस अ हेक्सोस शुगर थ्रू C4 pathway, what is the cost? बताओ. 30 ATP and how much? 12.
12 NADPH.
And to produce a hexose through C4 cycle, what is the cost? 18 ATP and
C3.
sorry, C3.
C3.
Yeah, C3 pathway. So did you get or not? ठीक है. So C4 pathway is more costlier, but we know C4 pathway, even though it is more costlier, but it is more efficient. This is more efficient. तो एक स्टेटमेंट लिख लो. At higher temperature, जो C4 plants हैं, at higher temperature C4 plants can fix 2 to 3 times more carbon. मैं क्या कह रहा हूं? At high temperature, this C4 plants can fix 2 to 3 times more CO2 in comparison to C3 plants. In comparison to C3 plants, they can fix 2 to 3 times more carbon. That means they are more efficient. They are more efficient, but at a certain cost. Is this clear or not? Okay. So we have seen C4 cycle, we have seen C3 pathway, C4 pathway, and we have seen photorespiration. So now do you understand? Do you understand why we need? What is C4? Why do we evolve C4? Just to take care of losses due to
Oxygenase activity of lubricant.
Is this clear or not? बात समझ में आ गई?
यस सर.
ठीक है. तो आज का लेक्चर हम यहीं समाप्त करेंगे. If you have certain doubts, you can ask me. जल्दी